Okay, well, it's my great pleasure to introduce Dr. Becky Roof from the NIH, NINDS. So Becky did her PhD with me at the University of Michigan in pharmacology, jointly with Hank Mossberg, a medicinal chemist in the pharmacy school. And she worked on a project of identifying and optimizing cyclic peptides to block the RGS G protein interaction. It was a design project where she took a piece of the G protein and used that to try to block the receptor, and we got some activity with that. And then she went on to try to optimize the peptides by making a 1B1 peptide library and doing an affinity isolation to try to figure out which peptides would bind more tightly to the G protein. After she finished her work in my lab, she moved on to Dave Sibley's lab at the NIH, doing D2 dopamine receptor high throughput screening. I believe, were you looking for allosteric modulators or just selected? Looking for allosteric modulators of the D2 dopamine receptor using high throughput methodologies. After she finished up with David, she worked in a couple programs at NIH before landing in her current position, which is program director in translational research, overseeing the IGNITE program. So with respect to drug discovery, I'd say NINDS is way ahead of most of the other institutes and has a tremendous number of opportunities, which she'll be telling us about here. So Becky, thanks for joining us virtually. I wish you could be here for real, but we understand the rules. And so welcome, and we look forward to your talk.
Thank you so much, Rick. I really appreciate that. Yeah, I really wish I could have been there in person too, but I apologize for that. But yeah, so today I'd like to talk a little bit about some of the opportunities we have at NINDS. We have an entire division dedicated to translational research, and we have a number of programs that I'll tell you a little bit more about. So first of all, I'll start with what our mission is for NINDS. I think most of you probably know that we're interested in seeking knowledge about the brain and the nervous system and using that knowledge to reduce the burden of disease. Now within NINDS, we have this division of translational research, and our mission there is to accelerate the application of that basic research findings and bring that to patients for neurological disorders and stroke. And we use funding, expertise, resources, not just grant money, to do that. So, like I said, we have a large number of programs. My goal is to make this not confusing to you because there's a lot here. I understand that you guys have a lot of drug discovery going on there at Michigan State, so I'm going to focus mostly on, let me go back, I'm going to focus mostly on IGNITE and Blueprint today. I'm going to touch on every program here though, because I want you to know what's available, and I want you to know who you can talk to if you have more questions about it. I also, of course, need to mention HEAL. So Congress has given NINDS, or NIH, I should say, $500 million to tackle the opioid crisis. Part of that is handled by NIDA and is around addiction, but part of that is developing better treatments for pain. And NINDS's point on that, of course, a lot of ICs have pain, so we coordinate across the board. But a lot of our programs have HEAL analogs that I'll touch on. I don't know if you have pain researchers or research going on at Michigan State, but this is, there's a huge focus on this right now, so I'll be touching on that as well. All right, and these are the programs, like I said, I'm going to focus mostly on IGNITE and Blueprint. I also understand you have some engineering at Michigan State, so I'm also going to talk a little bit about the device program. Now, stop me at any time if you want to hear more about any of these. Like I said, there's a lot here. We can do entire presentations on any one of these programs. So my goal is to kind of keep it at a high level, but please stop me if you want to hear more. All right, so starting with IGNITE, this is the program that I'm running, and I'm the point of contact for questions on this one. But IGNITE stands for Innovative Grants to Nurture Initial Translational Efforts. We have to have an acronym, so that's it. It is really designed to help people get to the later stage programs, such as Blueprint and CREATE, and I'll touch on those later. But some of the entry criteria for those later programs include things like having an assay that can support SAR, having a compound that you want to optimize, having proof of concept in vivo efficacy. So these programs here in front of you are designed to make that possible. So the first one, pharmacodynamics and in vivo efficacy studies. The second one is assay development and agent identification, which usually means screening. And then the third one is animal model development. Again, all of these are to get you the different pieces that you need to get to the later stage program. So for these ones, the budget is no more than $499,000 in any given year, and no more than $700,000 or $750,000 for the entire 3-year project. Our next application due date is June 19th. And I should mention, I'm happy to share these slides and send, if you have a listserv or something, I can send this to. This is a live link at the bottom. I tried to include that on all of the programs, so if you have this, you can find out more information. Now, Ignite uses an R61, R33 mechanism. So this is a mechanism that I think maybe a lot of people aren't familiar with. So I put a slide here to kind of demonstrate how this works. It is a phased mechanism, which means you have one phase, you pass a milestone, and then you get your second phase. So if you get your NOA, it will only be for the R61 phase, but then upon an administrative review, if your milestones are met, then you get your R33 phase. So the beauty of this mechanism is that it allows for dependent aims, which is something sometimes you have a hard time with in the R01 world. So if phase 1 is successful, then you move on to phase 2. And so we set this up so that the first phase is for demonstrating feasibility, getting your ducks in a row, and then in the R33 is the main event. So for example, you can use the first phase to develop your assays to do the high throughput screening, and then use the second phase to do the screen. So the reason we set this up this way is that when you go to review, you can say, these are the milestones we want. If we're successful in demonstrating that our assays are ready to go, then we move on to do the screening. And then reviewers are perfectly fine allowing dependent aims in that case. So it's really important here that the aims be very clear and quantitative, or the milestones be clear and quantitative to allow that this decision be made internally. It's an administrative review, it doesn't go back to peer review. All right, so I'm going to do just one slide on each of those FOAs so that you know what's involved. The first one for the pharmacodynamic markers and in vivo efficacy studies. Again, the goal is to demonstrate that your early stage therapeutics have the activity that would warrant further development. So you start with, it includes things like developing your pharmacodynamic marker, doing the study, doing PK, doing in vivo efficacy studies. And to come in, there are entry criteria. It needs to be something that's novel, that's a significant improvement over existing therapies. There has to be strong biological rationale. There has to be relevance for therapy development. All right. So I understand you have a screening center there, so this one might be of interest to your group. The assay development and screening FOA. So the goal is, of course, to develop new translational assays to screen and identify and characterize novel agents. The entry criteria, again, novelty, strong biological rationale, relevance for therapy development, and availability of agents or library. And then the third one, development and validation of model systems or pharmacodynamic markers. The goal, of course, is to promote a significant improvement in the translational relevance. So one issue in neuroscience is that sometimes the animal models are not what we would want them to be to support going forward, to support developing strong, therapeutically relevant therapeutics. So this is to help you develop animal models that would fill that gap, that might be more relevant or more useful for therapy development than what currently exists. So before I move on to Blueprint, are there any questions about Ignite?
So I just unmuted us. Any questions? Okay. Sounds like we can go
Okay.
ahead.
All right. So the Blueprint Neurotherapeutics Network, this program is pretty unique, I think, in that it combines grant support with contract resources. So we don't just give you funds in this program. If you get a grant in this program, you get the grant funds, but then you also get access to a lot of resources. It's really designed to help somebody who's developed something but doesn't really know how to go forward with it. We pair you with consultants, we pair you with contract research, contract organizations to do things like MedChem, to do things like IND enabling talks, all the way up to phase 1 clinical trials. So you don't have to have that expertise in your lab in order to move your drug forward, which is, I think, a really unique and really cool system that we have. So there's 2 FOAs here. One is a UG3 UH3, and the other is a U44 SBIR. So if you're not familiar with this mechanism, it's like the R61, R33, and then it's phased. But it's a U mechanism, which means it's a cooperative agreement, which means there's additional milestones along the way. It also means that there's more involvement with NIH because it's a cooperative agreement. And then this is the SBIR version, which is for small businesses. The next application due date is in August. Now, if you have questions about this program, Chuck is your point of contact. And it's also worth noting that this mechanism is a partnership among 9 different institutes, and INDS is the lead. So what this program supports is anything through exploratory, and by that we mean you have a compound, you have maybe a little bit of SAR, but you need to do more SAR. You should have a proof of concept by the time you enter at this stage. Hit to lead optimization, IND enabling phase 1. You can enter at any point, but wherever you enter, there's a 1-year preparatory phase. Now that's the first phase of the UG3 UH3. And that's where you can complete your SAR or your entry criteria for the SAR, the IND enabling studies, or whatever it is you need to do in that first year to just show that you are ready to go forward. So as this is an illustration of what I just mentioned, we will set up a lead development team that includes the principal investigator, consultants paid by NIH, and NIH staff. And this team actually meets every 2 weeks through teleconference. And we all together discuss what are the next steps, what compounds have been made this week and what do they look like and what compounds are we making next, for example. You have access for free to CROs for MedChem, data management, PK talks, clinical trials, formulation manufacturing. These are all through contracts. Now on the left here, I don't know if can you see my pointer? On the left here we have bioactivity efficacy studies. This is stuff that you do in your lab through the grant. And it's really important to point out that you as the PI will maintain intellectual property on this compound, or whatever you make out of this. NIH does not hold IP. None of these contractors will hold any IP. In fact, we had to do some contract gymnastics to make that happen so that none of the IP would be held in any of these contracts. So it's a really important point.
So yeah. Becky, I can mention for the group here, I believe do you know Mark Bailey? Mm-hmm. So Mark Bailey, who's a faculty member in our department, is the safety, one of the safety leads on the lead development teams for NINDS. So he has a lot
he's one of our
experience.
Consultants.
Yeah.
Yeah. Okay. Good. Well, he knows. Good. Then you probably know all about this program. You probably know all about this. No. Okay. Yes, that's right. He's one of our consultants.
So I have a question on the IP part of it, and it's really on the medicinal chemistry part. A lot of faculty may not have the expertise required to do the appropriate design. And how do you leave somebody off the patent from these companies, or do they just cede all rights and privileges? They're going to end up on the patent, no matter what.
Right. Right, so if AMRI designs the compound that is ultimately taken forward, they've signed it away. They've signed it to you,
Okay,
and that's in the
thanks.
contract. Yes. Yeah, and in fact, we have them on board because we know, the PI might not have that expertise. We try to make it, as accessible as possible for people to get things and move them forward. All right, so this program has been around since 2011, and it's been pretty successful. This slide's actually a little bit out of date. We have two new projects that haven't been added to this. But as you can see, we've had a number of different projects come through and a number of different disease areas through a number of different institutes. I'm going to point out all of these money bags, these green things here. These are situations where the applicant has been successful in using what NIH has supported them to do and have gotten outside licensing or an outside investment deal that's been publicly announced. So there's a lot of these, and we're pretty proud of that. And I think, it suggests that, we're putting this together in a way that's helping people, and that's really our goal. So we also need to point out, on the other hand, well, not on the other hand, it's just the way it is. There are projects that don't continue. Since this is a milestone-driven program, although some of them that haven't continued have still gotten investments. So we've still been able to de-risk something or bring it forward enough to the point where investors are interested, even though the science may not have supported them going forward in the particular direction that they had gotten the grant for. So we're pretty proud of this program. Right. So this is a biologics program that's in sort of the same space as the Blueprint Neurotherapeutics program. It's but it's for biologics, peptides, proteins, oligonucleotides, gene and cell therapies. But again, it's the same space. Once you graduate Ignite, you can use Ignite to develop these biologics. Once you have your proof of concept and you have your path forward, then you can move forward into this CREATE biologics program. I'm not going to spend a lot of time on this because I understand you're more drug development focused there, but I wanted you to have this information. And the point of contact for this one is Chris. All right. Now, this is one of the HEAL FOAs. So a lot of our DTR programs have HEAL analogs because we have this $500 million that NIH has that we need to spend. Not all of that, of course, is in NINDS or in pain, but we have separate programs to help us spend some of that extra money. If you are doing pain research to develop something that is better than the current, something that's non-addictive, better treatment for pain, and you're in the Blueprint or CREATE space, this FOA is something to look at. There's, again, an academic version and a small business version. And if you're doing small molecules, Chuck Simon is your point of contact. And if you're doing biologics, Chris Boshoff is your point of contact. All right, so back to our programs. So I've told you about Ignite, I've told you about Blueprint, told you about CREATE, and then also these HEAL versions. Next, I'm going to tell you a little bit about the biomarker program. I'm not going to go into a lot of detail on this one, but stop me if you have questions. All right, so the goal of this one is to facilitate the development of quality biomarkers for neurological diseases to improve the quality and efficacy of clinical trials and clinical practice. So the point of contact for this one is Marianne, and her information is down here. Now there's 4 different sets of FOAs here. The top ones are NINDS for any NINDS indication. The bottom ones are the HEAL versions for pain indications. One important point about all of these HEAL FOAs is even if you're doing a pain indication that is not traditionally supported by NINDS, if it's under the HEAL umbrella, we can support it. If anybody has questions about that, I can get into that in more detail, but that is an important distinction for us. Oops, I'm sorry. All right, so for the NINDS biomarker programs, there's a first one for analytical validation of a candidate biomarker, and then there's a clinical validation program. And then in the HEAL, we have a discovery program and an analytical and clinical program. So they're a little bit different, but still towards the same goal. All right. Now,
So Becky, before you leave the biomarkers, what is the scope of biomarkers that might be considered? Do you have a sense of what they're looking for, blood or functional, or what kinds of things?
You know, it can be anything, right? So blood is one, imaging is, another big one for, the neurospace, but really it can be anything. It can be functional. Yeah, I don't think there's any really limitations there as long as it's something that's going to be translationally relevant or will really help in a clinical design or in patient use. So I think that's really much more important than the modality. All right. So now I'm going to touch on the device program. I understand you have engineering folks there. Now, I admit this is a program I know least about, but I have your point of contact here, Nick Langles. He can tell you all of the details. Now, there is a whole suite of programs for device development going, from early stage concept generation all the way up through clinical trials. There's a whole series of these. NINDS works closely with other institutes, even NSF here. Some of these are run by NIBIB, which is the National Institute of Biomedical Engineering, on a lot of these. So, these are all partnerships. But of course, the goal is to support development, optimization, and clinical and translational activities of small clinical studies for therapeutic or diagnostic devices. Okay, so I'm going to talk about just, I don't know if you can see my cursor, but the Translational Neurodevices program. And then I'm going to touch on a HEAL version of this. So this is one that is run by NINDS. I believe other institutes are involved, though. For therapeutic and diagnostic devices, it's again a cooperative agreement. There's an SBIR version, as well as a academic UG3, UH3 version. But the program scope is really for very close to final system and manufactured using close to the same manufacturing process as what will finally be used in the larger clinical trial. If you're not at that stage, there's those other programs that I just showed you to get you there. And it does require early feasibility clinical data to inform the final device design. Again, there's the UG3, UH3 version, and the U44 version. All right, and then with the HEAL, again, there's more FOAs with the HEAL because, that's a separate program. This HEAL program is integrated with other, not just the NINDS program, but there's the BRAIN program. I don't know if you're familiar with that. Brain research through advancing innovative neurotechnologies, which is a whole other big multi-IC suite of programs. There's also the SPARK program, which is another multi-IC suite of programs, and these really all get integrated. Again, if you have, so this is a common fund program, but if you have questions, Nick Langles is your point of contact for that. All right, so how are we on time? We're good. So I've covered the ones that I was asked to focus on. I'm going to just touch on some of the other programs here, but stop me if you have questions. All right, so back to HEAL, helping end addiction long-term. I'm not going to go through all of these, but I'm going to point out that there's this whole suite of programs. I've already talked about drug development, and the devices, I've touched on those. One thing that I will talk about next is there's a, actually go there. There's a screening platform. So if you're interested, this is something that's just getting set up. It's a free resource. We will screen your compounds for you in various pain models and give you the data. Totally free. It's the goal is, and not just your molecules, but biologics, devices, natural products. The goal really is to create, you know, a larger data set that's available for people. We'll screen it for your testing in pain animal models, but we'll also screen it for new receptor binding. We'll look at acute and chronic pain. We'll also do in vivo addiction screening. So this is really just to help people get things moving forward that could potentially be better pain therapies. Likewise, we have an epilepsy screening program, which is another freely available screening program. This one actually existed long before the PPSP did. It's been around since 1975, and it's actually contributed to a number of currently used anti-epileptic drugs. Oh, here you go, a role in 9 marketed treatments for seizures. So this has been really successful, and it's freely available. It costs you nothing. You just send in your compound. Talk to John Keene about this program. All right, the small business program. So if you develop something and you'd like to spin out a company around it, we have a program for that. Congress mandates that we spend 3.65% of our R&D budget on small businesses. And not just NIH, but any federal agency with a substantial R&D spend has to dedicate a percentage of it for small businesses. And it's really for any kind of R&D with the potential for commercialization. And it supports not just therapeutics, but diagnostics, research tools, anything from bench research to translation to early clinical studies. There's a lot of funding opportunities. A lot of the programs that I've talked about today have SBIR versions. So if that's something that you can utilize, if you're working with a company, if you're spinning out a company on your own and you're not sure if you should go that small business route or the academic UG3, UH3, or R1 route, the SBIR route is usually the better way to go because it has set aside funds. So that's something to be aware of. Oh, I'll also mention with this one with HEAL, we're required to spend 3.65% of our HEAL funds on SBIRs. So even though there isn't a HEAL SBIR FOA at this point, we're still very interested in anything that would be applicable for pain or addiction in SBIRs. Okay, all right. This is the last program I'm going to talk about. I'm going to be quick. Countermeasures against chemical threats. I don't know if anybody's interested in this or if this resonates with anybody, but there is a program for helping us come up with, treatments or antidotes to any kind of chemical threat that might occur. All right, so I want to leave lots of room for questions. So I'm going to kind of close up here with general words of advice, and then I'm open to any questions you might have about anything here. It's always good to contact us in advance. You know, I know a lot of time and effort goes into putting applications together, so if we can help you make sure that you're in the right FOA or that what you're proposing is within mission for NINDS, you know, that's what we're here for. Also, we listen to a lot of reviews, so I often tell people, you know what, the review's been really hampering on this lately, make sure you explain that. Like that kind of advice, I think, you know, we try to give. So contact us in advance, carefully read the FOA. A lot of these are different mechanisms than what people are used to, so look at it carefully and ask us if you have questions. If milestones are required, they need to be really clear, they need to be quantitative. You're not going to be successful if you say, you know, this is the list of activities we're going to do. and that's it. Or, you know, we expect our compound's going to be fabulous, and then we're going to go forward without quantitating that or explaining that, or even if you are very quantitative, why did you choose the cutoff you chose? That's probably, like in my experience with Ignite, that's one of the biggest hiccups people come across when it comes to review. It's just something that people aren't always used to thinking in those terms. Rigor is something NINDS is extremely important, interested in. We've had a notice since 2011 about the importance of rigor and reproducibility. We ask our reviewers to intentionally look at that question and comment on it in the review session. So please make sure it's there. It's a very preventable problem if reviewers come up with issues there. Multidisciplinary team is something that's important for a lot of these programs. A lot of these programs require that you have, you know, a biological expertise, but also translational expertise and whatever is required for bringing your particular product forward. So multidisciplinary teams are really important here. Discuss intellectual property if you're developing a therapeutic. You don't have to have IP in place, but it's really helpful that to show reviewers that you've thought about the issues, that you thought about how you're going to take it forward, that you've thought about the steps. It doesn't have to be in place. You can just have a letter of support from your tech transfer office saying, I'm happy to work with you. Like that's usually good, at least for early stage anyway. And these are translational applications. So discuss how this will translate. What are the next steps? You have to be a little bit more forward thinking in translational programs than you probably do in regular, basic science R1 space. All right, so with that, I'm just going to leave you with, yeah.
Quick question, if you don't mind.
Yeah.
If you go back one slide again.
Mm-hmm.
So intellectual property comes in a number of different flavors, right?
Yeah.
Some include composition of matter, some might be use. Is there a particular, what are you looking for in particular there?
There isn't one answer. It kind of depends on what you're doing. The key really is to illustrate that you have a path forward. And whatever that is and whatever's applicable to your particular situation is and whatever you can convince reviewers about is really the better way to go.
Okay, thanks.
Any other questions? I left lots of time for questions.
Okay, well, so you have one more slide, it looked like.
Just
final
links for more information.
Sounds good. So we can open this up for any questions people have now.
Yeah.
Several.
So for the Ignite program, what?
Actually, could you introduce yourselves when you ask?
Sure. Patrick, I'm a grad student here. For the Ignite program, what kinds of preliminary data are reviewers looking for?
Are you thinking about a particular FOA?
Just in general.
Just in general?
Compound screening or what? So the Ignite had 3 different things, right, Becky?
That's right. Do you want me to go back or?
I think assay development was one of them, right?
Okay. Yep. Yeah. So some feasibilities, you don't have to have things obviously all your ducks in a row because it's for development as part of the FOA, but some indication that it's feasible to do what you want to do. Some people come in with, we have this assay, but we need to miniaturize it, or we have one assay, but we need to develop a secondary, tertiary assays. Some people don't even have that, but have reason to think that it's feasible to develop. It's not really a hard rule there. Also, evidence that the target you're after is worthwhile.
I have a question about the, Jets Tepian, Department of Chemistry. I have a question about the Blueprint program. In terms of repurposing of compounds, is it a advantage or is it maybe a turn-off to have a medicinal chemistry team repurpose compounds for a different target?
Right. So if you're having a chemistry team, I'm assuming are you starting, is that your starting point, but then you're changing the structure and you're developing something new?
The molecule in order to select for one different target than the original molecule was meant to target.
Yeah, well, so as long as, I wouldn't say it's necessarily a turnoff. I mean, it means you're farther along probably. You would need to have evidence, some kind of evidence that the new target is worth, that makes sense for, that this would work on the new target. But yeah, if you're, if you still need to do the medicinal chemistry and stuff, then that would be appropriate.
So that gets back partly to Edmund's question on composition of matter versus use patent. So I think whenever you're repurposing, there's always a question of IP and how you take that forward.
Yeah. Yeah. I don't know if it's necessarily a negative, but you would definitely need to convince reviewers that you have a path.
So one question I had is sort of I guess, operational, but Tom Dexheimer, who runs our high throughput screening core, was at NCATS, and their screening center for 5 years before we got in here. How much do you work with NCATS in these various programs in terms of the screening centers there as a resource?
We don't work directly with them. I've had a couple conversations, but if you wanted to use their screening center and then have a grant with us, we can't pay NCATS. We can pay you, but there is that. So how you work that out is between you and NCATS.
Interesting conundrum.
Yeah, but I mean, you can work with them. Certainly, you can work with whoever you want to, and they have resources.
But you have to convince them to put their resources toward your project.
Right. Well, and I would ask them how they handle that for any grant, because anytime they're working with academics, I would think they would run into that.
Yep. Okay. Other questions? Yeah. See.
This is Sing Lee from who is also in this department, and I'm wondering if you can provide more information about the Create Bio, the program related to biologics.
Okay. I can go back. All right. So to tell you a little bit more, this program, they have, it's not quite like Blueprint where there's a whole large suite of contracts that are available to you, but they do have a number of consultants that are available. But aside from that, the work has to be done in your lab. They have 2 levels where you can come in. You come in at the optimization level or the development level, depending on where you're at. The development level is farther along. The optimization level is, a little bit earlier. So this is where you would be if you have your proof of concept, you have either your gene therapy or your peptide or whatever that still needs some tweaking or maybe some optimization or isn't quite ready to go. Here, you're more solid, you're ready to go. But the goal really is to have something that's at the end ready for an IND or even a phase 1 clinical trial. Is there particular more information you want? I didn't answer your question.
So one thing, Becky, you mentioned earlier also is that Ignite can work on biologics projects, and yeah. I don't know what stage you're at saying, but it may be that a biologics project through Ignite might be a more appropriate entry point for that.
Yes, that's right. Yeah, so Ignite is a feeder program for both this and Blueprint. So we support peptides, proteins, oligos, gene cell therapies, in addition to small molecules.
Thank you.
Any other questions? Okay, well, let's thank Becky for a great presentation. So I have seen that you have a very busy schedule with a lot of teleconferences coming up.
Mhm.
And I think all the people who were on those have gotten an email about the timing and all that. The first one with Min Kuo is actually, he's over in the other building and I saw him online. So
Okay.
he'll be there. If there are other people who would like to meet with Becky, I don't know what your schedule is. If we added someone at the end or
Mhm.
had squeezed someone in now, would that be okay?
That would be fine. Yeah, as long as I have an appointment at 4, so until 4, I'm open.
Okay, sounds good. So if anyone is interested in speaking with Becky who didn't get on the schedule, let me know, and we can arrange to get that all set up. You know, there is a little bit of time now before the next talk. If any of the students would perhaps like to, you know, talk to Becky about opportunities, students, trainees,
Oh, yeah.
maybe we could do that, you know, now,
Sure.
while you're waiting for Min's time to come up.
Okay.
Very good, thank you. So any students wanna stick around? Okay, I guess you have 45 minutes until Min's presentation. Very good, and I'll be talking to you later.
Great, thanks.
So, Becky, it looks like someone was interested in talking to you.
Jake.
Oh,
Well, she doesn't have
sure,
any
sure.
She has
Yeah,
ample.
why don't you just take over the room and, I'm sorry, yeah, there's control.
So actually,
So,
while
okay,
like
very
Anna's
good.
data suggesting.
Okay, there you go.
Related to
I'll talk to you later.
Because if you add and
Hello.
eggs list.
Let's move
Yeah,
out.
we need
Let's
to
go
move
talk
outside.
somebody else.
Can you hear me?
Email.
Can you hear me?
It might be, error on our.
Can you hear me now?
I can hear you, yep.
Okay, yeah, my name is Yasser Al-Damin. I'm an assistant professor at microbiology and medical genetics. So I submitted an R1 on October, and
Okay.
for that I proposed to develop a mouse model to study neuroinflammation. So I study a specific immune receptor, and we published so many papers around that story. But I'm immunologist. I'm kind of new to neuroscience, and in that grant, I proposed to develop condition knockouts, so specific condition knockouts in microglia
Okay.
and some other immune cells, and they specifically asked me to go and develop the model in order to, at least for my grant to be reviewed. So for the Ignite program, so what do you think? Like I'm in the middle now of developing the condition knockout. So I have the LOXP and gonna acquire the pre mice and try to breed them. So obviously this
Mhm.
takes time. It's gonna take like another like 4 to 6 months just to get the mice. So can I, like with what I have right now, can I still apply for the Ignite program, or I need to wait longer, get the mice, and then apply? What do you think?
Well, so, couple of questions though. What disease indication are you, the goal is to develop therapeutically relevant animal models. So what disease would this be useful for? How would this be
Um,
therapeutically, useful for developing something?
Sure, it's gonna be for MS,
Okay.
and yeah, and possibly for other neuroinflammatory disorders as well. But in the proposal, I specifically mentioned EAE like using the EAE mouse model to understand the role of this specific protein in multiple sclerosis.
Okay, so your goal is to understand the role of a particular protein. That might be a hard sell. So the goal, if the goal is really to understand the basic biology of the disease, that's not a fit for the FOA. It's really assuming we have a basic understanding of the biology, we're going to apply that to something that we will then use that's ready for therapy development. If you're still understanding the role of it, then it's hard to argue that it's ready to be, you know, a therapeutically relevant target. Does that make sense?
Yeah, what I meant is like I already developed, so we have patents. I'm in the process also of developing a small molecule to target that
Okay.
protein. So it's an immune receptor in a human, in all immune cells really, but we published data showing that we can block type of interferon responses by just activating the receptor. So I also developed an FC fusion protein, so biologics, that blocks the receptor and show that it can exacerbate the pro-inflammatory state. So, so basically, like activating the receptor blocks multiple cytokine pathways in response to certain TLRs and type of interferon receptor signaling. So now what I'm trying to do is develop a conditional knockout mouse model and show that this protein is relevant for neuroinflammation, with the goal of developing a small molecule that can activate the receptor in order to block neuroinflammation in multiple sclerosis or other neurodegenerative diseases.
Okay. Right, so you're knocking out this receptor, but then that receptor would be the target for future therapy?
Yes, yes.
Okay.
So, yeah.
So how would you use that knockout animal then to develop a therapy? Or is it
I
really
will
to
use-
understand the role?
Yeah, I will use it as a proof of principle to show that this protein has a relevance in neuroinflammation. But of course, the goal is to develop the small molecule or the biologics and then use the wild type animal to develop the data. So we already published data using human monocytes and T cells, but now I'm going back to animal in order to develop kind of a proof of principle in vivo data.
Yeah. Okay. So it's sounding to me like this is really to get at the mechanism of the disease and not really something that will be used to develop your therapeutic. I mean, it's a basis for that. It's an understanding of that, for that.
I see.
So again, that FOA is really not for understanding the disease. It's for this model will be better suited for developing a therapeutic than maybe the EAE model is, for example, in your case. Does that make sense? Yeah.
Sure. So you think then this should be like an RO1 route than Ignite?
Yeah, it's sounding like it.
Okay, yeah.
Okay.
I think that's what I wanted to know. Perfect. Thank you very much.
You're welcome.
Thanks.
Thank you.